Hi everyone, thanks for joining today. We'll get started in about 5 minutes. Alrighty. Hello everyone, and thank you all for joining today. Welcome to the NOAA Omics seminar series. I am your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted on our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation, and at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Grease Under Pressure, What Lipidomics Can Tell Us About Adaptation Throughout the Water Column by Dr. Jacob Winnikoff. I'll go ahead and share screen sharing permissions. Jacob Winnikoff is a marine biochemist studying adaptation to extreme environmental conditions. Jacob grew up in and around the temperate waters off Southern California, and as an undergraduate, he studied the heat tolerance of intertidal mussels in George Samaro's lab at Hopkins Marine Station, earning his BS in biochemistry and biophysics from Stanford in 2016. His graduate work focused on high-pressure adaptation in comb jellies and took place in Stephen Haddock's lab at Minbari. It concluded in 2022 with a PhD in ecology and evolutionary biology from UCSC. Jacob was then awarded a NASA postdoctoral fellowship to investigate microbes' tolerance of harsh environments on other ocean worlds. He currently works as a postdoctoral scholar in the Giras lab at Harvard on projects involving lipidomics, proteomics, and metagenomics in such diverse marine systems as hydrothermal vents and diving mammals. Thank you, Jacob, for presenting today. And I will now pass it off to you. We can see your first slide just great.
Okay, thank you, Nicole. My keynote's being a little bit touchy. I'm trying to make sure you can see my pointer on the screen. But good afternoon to everyone here. Thank you, Nicole, for organizing and for the introduction. Thanks to Zach for the invitation, and thanks to you all for joining. Here's how I intend to spend our next 40 minutes or so. I want to leave ample time for questions and discussion. I'll start by way of an introduction explaining what lipidomics means and trying to convince you that lipids are just as cool as DNA. Then comes the juicy science part of the talk, where I share a story about lipids' role in deep sea high pressure adaptation. The spoiler alert is it involves a comparative study with ctenophores, also known as comb jellies. And then finally, I'll quickly share a code resource for interpreting lipidomics data in ways that can tell us about an organism's physiology. So here we go. So what exactly is lipidomics anyway? Here's how I think about it, just to get you oriented. There's all of omics, which Nicole outlined nicely for us a minute ago. Your more mainstream omics are the ones you use a sequencer for, genomics and transcriptomics. Can you see my mouse, by the way, Nicole? Or no, my mouse pointer?
Yes. Yep, we can.
Wonderful, thank you. Then there are other omics you use a mass spec for. So they're basically sorting molecules based on their weight and quantifying them. I put proteomics in this intersection here because you're actually using a mass spec to work out an amino acid sequence. And then you've got all of metabolomics. So these are the products made by the proteins encoded by the genes. And there are a whole bunch, but when we focus on the lipids or fatty molecules, we call it lipidomics. And here's what lipidomics can tell you about an organism. Basically, two things of general interest. It can tell you what it's eating and how much. You get this info from storage lipids like triacylglycerols, which I've cartooned on the right. And this is not my main interest, but I'd be happy to circle back on that application of lipidomics in Q&A if you're curious. And then lipidomics can tell you about an organism's environmental tolerance, specifically with respect to temperature and pressure, which are very relevant parameters in a changing ocean. And you get this information from phospholipids, which is the cartoon here. Phospholipids are the building blocks of membranes. They're connected to environmental tolerance because they form cells' interface with the outside world. I'm guessing that the phospholipid bilayer rings a bell for most people here. And phospholipids' behavior is very sensitive to temperature and pressure. I'll spend the next few minutes explaining what I mean by this. But first, a really quick introduction to these molecules that are going to be the star of the show. So phospholipids are amphiphilic, meaning that they have both water-loving hydrophilic and greasy water-repellent hydrophobic parts. The water-loving section is this phosphate-bearing head or head group, and then the two hydrocarbon chains are greasy hydrocarbons. The actual chemical structure looks like this. There are about a dozen different head groups you can tack on. And then the hydrocarbon chains can vary a lot in length. In other words, they can have different numbers of carbon atoms. They can also contain double bonds. And each one of those introduces a physical kink to the chain like this. Another word for the number of double bonds is the amount of unsaturation. So if you've heard of polyunsaturated fats, those are the ones that are good for your brain. They have a lot of these double bonds. I've mocked up this phase diagram in the middle, which I'll explain, to show you exactly how temperature and pressure affect the behavior of lipids. So keep in mind that for this whole example, I'm not changing the lipid chemistry at all. I'm holding the structure of the lipid molecules constant and just tweaking the temperature and pressure. And the way the phase diagram is shown, temperature is increasing from left to right, and pressure is increasing downwards, just like it does in the ocean. Each color here in this rectangle in the middle denotes a distinct phase or an arrangement that the lipid molecules can take on. And by the way, these three colors here are going to mean the same thing all through the slides. So for the most part, living membranes in an organism are in this beige liquid crystalline phase in the middle, where you've got phospholipid molecules that are arranged in a bilayer, tail to tail, the greasy parts stick together, but they're constantly dancing around each other sideways. Within this phase, a higher pressure or colder temperature down and to the left will make them move slower. It makes the membrane more viscous, oozier, and thicker. Conversely, warmer, lower pressure will make the lipids dance faster and the membrane gets thinner. Membrane viscosity actually controls cellular respiration. So liquid crystalline membranes have to operate in a sort of Goldilocks zone, not too viscous and not too fluid. At the extremes of high pressure or low temperature, that's the lower left here, the lipid molecules actually stop dancing altogether. They lock together into what we call a gel phase, and that's prone to breakage and leaking and stuff like that. Obviously problematic. Conversely, at extremely low pressures or warm temperatures shown in pink, the lipid molecules don't actually stick together in a membrane anymore. They peel apart, flip inside out to form these tubes. These are, this is sort of a cross section, but imagine these as tubes of lipids sticking out of the screen at you. And we call that an inverted phase because it's inside out. For obvious reasons, this arrangement won't function as a barrier at all. Just before we press on, I want to emphasize that it's tempting to look at that lipid behavior and think that a liquid crystalline phase is good and a gel or inverted phase is bad. But the picture is not quite that simple. Multiple lipid phases, arrangements, actually have to coexist in the living cell for it to function. So membranes in the living cell are mostly liquid crystalline, beige, but then they have these gel-like lipid rafts in them. And then actually for cells to divide or for compartments to bud apart from a membrane or fuse to it, the membrane has to go through this transient inverted phase. So the cartoon on the right shows two membranes coming together and what that looks like. And what that means is that the borders between all of these membrane regions in a single cell have to balance right on the cusp of a phase change, even though the environmental temperature and pressure might be changing. So living cells pull this off by regulating their lipid makeup. By changing small features in the lipid chemistry, a cell can nudge its membranes back and forth across those phase boundaries. So shorter hydrocarbon chains and double bonds, remember they create these kinks, they prevent the lipids from packing together in a really organized way. And that means that the membrane becomes more fluid. You get a liquid crystal. These lipids are actually pretty chemically similar to what you see in olive oil. And when you think about olive oil, it stays fluid down to a pretty low temperature before it gels. Longer straight chains behave more like butter. They set into a gel very readily. And then, I'll get to that in a second. Head groups that are small give a lipid a cone-shaped profile like this. They cause the sides of the membrane to peel apart, and that ends up forming an inverted phase. Conversely, head groups that have a cylindrical profile let the membrane lay nice and flat. Okay, so what is this big word that popped up on the left prematurely? There is actually a name for this particular form of adjustment, which basically keeps the lipids dancing around each other at just the right speed. It's called homeoviscosity, referring to homeostatic control of membrane viscosity. Viscosity, by the way, is related to fluidity. They're basically opposites of each other. And we've known about this thing, homeoviscosity, for 50 years. It happens as a form of adaptation in everything from bacteria to reindeer hooves. Maybe some of you have heard the term before. But we've got a whole like 7 minutes without mentioning the ocean, so let's bring it back. We know that life uses homeoviscosity to deal with temperature differences, to adjust the fluidity of the membrane. In the ocean, there are temperature differences, but there's also pressure to contend with. There's 1 bar, 1 atmosphere of pressure for every 10 meters depth. You all know now that cold and high pressure both push membranes toward this lower left gelled up corner of this phase diagram. But the question we wanted to ask is, does evolution deal with cold and pressure in the same way? Is it all homeoviscosity? Can you just prevent the gel phase and you're okay? Or is there something else at play? That's the big question behind the research story I'll share today. And to answer a question about adaptation, we needed a comparative system, and we chose ctenophores. If you're curious why we picked ctenophores, feel free to ask afterwards. But for now, what you need to know is that they're gelatinous animals. They're also called comb jellies for these comb-like rows of tiny paddles they use to swim around. And comb jellies actually make up their own phylum. They're completely distinct from your cnidarian true jellies, which are at the bottom of the tree here. In fact, we, the bilaterians in the middle, are more closely related to cnidarians than ctenophores are. So back to our question about lipid adaptation. To start answering it, we sampled a whole lot of ctenophores across a diversity of depths and temperatures. So after a couple of years of ROV, scuba, and freezer diving, I managed to gather 105 individual ctenophores across 21 species. And they're all plotted in this depth temperature space. Depth is increasing downwards again. To separate the effects of pressure and temperature as much as I could, because everybody knows water gets colder as you go deeper, I ran analyses within slices of the depth temperature space. So for depth analyses, I held temperature fairly constant below 7.5 Celsius. And for temperature analyses, I held pressure fairly constant, shallower than 200 meters, less than 20 atmospheres. The specimens in the upper left corner, they're all from the Arctic, and they're really critical to the study because they provide a common reference point for both the depth and temperature comparisons. I extracted lipids from all 105 animals, and then I cut the hydrocarbon chains off the lipids and analyzed them in bulk in a gas chromatograph mass spec. This is conceptually a little bit like barcode sequencing. You don't get the whole story, but you get some useful info and you can afford to run a lot more samples. So you'll remember that the legs of the lipids can vary in length and in the number of double bonds. And those features control membrane fluidity. So here on the left, I've plotted length and double bonds on the y-axis as a function of temperature and of depth. Each data point is an individual animal. And what we can see is that as we go colder, the chains get shorter, but the number of double bonds doesn't really change significantly. And conversely, as we go deeper, the number of double bonds go up, but the length of the chains doesn't change that much. And that all makes good sense because adding or making the chains shorter as it gets colder counteracts the gelling effects of colder temperature. The lipids don't stick together as well if the chains are shorter. And adding the double bonds prevents the lipids from being packed together by pressure. But what is really clear is that evolution is not dealing with cold and with high pressure in the same way. It's using different biochemical strategies. So we've answered our question, but next, of course, we wanted to know why. And the lipid legs alone weren't going to tell us. We had to do some proper lipidomics. That means keeping the phospholipid legs attached to the head and quantifying them with or liquid chromatography mass spec, LCMS for short. This is the kind of data we get. I just want to sort of highlight its complexity, the richness of the data here. Each column represents the phospholipidome, all the phospholipids, in an individual ctenophore, and each block here is a distinct lipid. So you can see the lipidomes are quite complex. When you think about it, you've got a dozen different lipid heads to choose from and about 30 different hydrocarbon chains you can attach. So you combine them all, and in the end, we saw 995, almost 1,000 unique phospholipids across these 5 species. Here I've averaged the 3 animals in each species and combined the lipids under each head group type, which are shown by color, just for clarity. And I want to point out that the species are arranged in a particular order from left to right. They go from warm to cold at roughly the same depth. These are all shallow animals. And then from the middle to the right, shallow to deep. And the deepest animal here lives at about 4,000 meters, 400 atmospheres of pressure. But because the one in the middle lives in the Arctic and the other 2 live in the deep sea, they all live at around the same temperature. If you start to squint at these color blocks, you might be able to pick out some interesting patterns. One that really stood out to us was one in this head group class called plasminal PE, or PPE for short. It increases about 6-fold from the surface to 4,000 meters deep. That's the center to the right. But in colder water, that's moving from the center to the left, it also increases. It increases. Sorry, in colder water, going from the left to the center, it decreases. So that doesn't really make sense if it's just controlling membrane fluidity, because it's actually responding in the same way to high pressure and high temperature. So it must be doing something else. And just to prove that this depth trend is a thing, here's the full data set. We ran lipidomics on a lot more species. And I've just plotted PPE percentage, basically, mole fraction versus depth. You can see that at 4,000 meters, it makes up more than half of all phospholipids. But we still didn't know what it was doing. So hold that thought for a minute. At the same time as we're doing all these lipidomics, we're trying to get a handle on how ctenophore membranes actually behave biophysically at different temperatures and pressures. One of the ways we did that was with this other technique called X-ray scattering. And it works like this. You extract the phospholipids from a ctenophore, and you mix them with water, and they actually form membranes all by themselves. It's basically because oil and water don't mix. So the hydrocarbon chains stick together, and the head groups face outward at the water. You make these artificial membranes, you cram a bunch of them into a tiny tube, and then you shine really powerful X-rays through it onto a detector. And generally, to get X-rays this bright, you have to visit a synchrotron particle accelerator. There are a few of those available around the country. The type of bullseye pattern you get made by the X-rays on the screen tells you what arrangement, in other words, what phase the phospholipids are in the tube. And the coolest thing about the facility we used at Cornell is that you can actually apply deep sea pressures to the lipids. So here you can see the lipids going from an inverted phase at room pressure to a liquid crystal membrane when you compress it, and back again. So basically, using this tool, we can generate real versions of the phase diagrams that I showed you in the intro. And we decided to try that with 3 ctenophores to start. Here are the first 3 we looked at. They're in the same order as before: warm to cold from left to middle, and then shallow to deep. Here I've highlighted where each one of them lives in the pressure temperature space. So I've got the same coordinates here. Pressure is increasing downwards. By the way, I'm not asserting that we know exactly how these animals' membranes behave in vivo, but I think the differences between species that I'm going to show you here are valid. So for the shallow temperate species, this is from shallow in Monterey Bay, we see this narrow fluid bilayer region near its native conditions. but if we compress it, it gels. If we heat it up, the membranes flip inside out. When we compare it to the Arctic shallow species, the most noticeable difference is that this gel phase boundary here has moved a ton. It's way up around 800 bar, which corresponds to a depth of 8 kilometers that, by the way, this species never sees. But the comparison between the shallow and the deep species is the one I really want to highlight. They live at similar temperatures because this one's in the Arctic, but this one lives at 1 atmosphere and the one on the right lives at 400 atmospheres. The gel transition stays in essentially the same place, but the inverted boundary here is at a much higher pressure. And this is remarkable because the inverted phase tends to be really unstable when you squeeze it. You can intuit that just by looking at the cartoon of these inside-out lipids. There's this void region in between the tubes, and it's very easily crushed. So really unusual, striking behavior that it can stay inside out like that to a high pressure. There's nothing in the tube but lipids and water. So we knew that something in the lipidome was driving that behavior. And naturally, we wanted to know, coming back to our lipidomics, if it was our friend PPE, that deep sea lipid that goes way up with depth. We ordered some of the pure stuff and ran more X-ray experiments with it. Here we're holding the temperature constant and just pumping up the pressure from left to right in these plots. And we compared PPE with its sort of closest structural relative, which we just call PE. And the difference between ordinary PE and PPE is actually just that you take away this carbonyl oxygen and you add a double bond right there. Very small difference. But we saw a very, very different behavior. And the effect seems really similar to the pattern we saw in the animals. We can pump up the pressure on the PPE, but this inverted phase arrangement doesn't go away completely until about 1,000 bar. By the way, we tried this with 2 kinds of hydrocarbon chains just to make sure that the head group type was what was actually having this effect. In addition to figuring out lipid phase, you can also use X-rays to measure the approximate shape of a lipid. You can tell basically how tapered the shape is, and that's what I'm showing on the y-axis here. Down at the bottom of the y-axis, the lipids are super cone-shaped. At the top, they're more mildly tapered. And you can see that as you pump up the pressure from left to right, both of these lipids, the PE and the PPE, become less tapered. But the thing is, the PPE is always more tapered at every pressure than the PE. And if you compare them, it makes up for the effect of about 1,000 bars of pressure. So it looks like that is why it forms an inverted phase. Because PPE is tapered, it makes the two halves of the lipid bilayer want to peel apart, and it forms that inverted phase. So what it was starting to look like to us was that pressure and the molecular structure of PPE have strong opposing effects on lipid shape. Pressure wants to make lipids less tapered, and including PPE in the lipidome makes the lipids more tapered. They work against each other. And that got us thinking that maybe lipid shape is what evolution really, quote, cares about under pressure. Conveniently for us, membrane biophysicists came up with a number to describe the shape of membrane lipids, and they call it intrinsic curvature. Inconveniently, that number feels sort of backwards. So lipids with negative curvature are shaped like traffic cones, like PPE. Lipids with 0 curvature are perfectly cylindrical and they form a flat membrane. And then lipids with positive curvature are sort of ice cream cone shaped. Better yet, those same biophysicists who came up with that number have measured values for curvature for pretty much every major lipid head group, except for PPE, and we measured it for PPE. So what that meant is that we could do this transformation where we measure the lipidomes with mass spec, like you saw earlier, and we multiply every component, basically every color block here, by its respective curvature, and we get a sort of weighted average, which approximates the curvature of the entire membrane lipidome. Mind you, it approximates it at atmospheric pressure because all these values in the middle were measured at atmospheric pressure. The plot on the right shows how the different components contribute to the lipidome curvature. So the positive components go up on the left for each species, and then the negative components come down on the right, and you end up at the bottom of that green bar with the total average curvature for that species. You can see that the PPE, the light green bar here, is doing the lion's share of the work in all of these ctenophores. When we calculated that index for all 17 species, even the ones that were living at warmer temperatures, we got this strikingly strong trend. So it looks like adaptation is working against the effect of pressure so that lipids stay tapered even at depth. So each of these dots here represents a different species of ctenophore. And on the y-axis, it shows how tapered their lipids are at atmospheric pressure. The deeper ones are more tapered, but you can imagine that when you squeeze them, the taper ends up being about the same as the shallow ones have at the surface. We decided to give this adaptive strategy a name. And the hypothesis we came up with, we call the homeocurvature hypothesis. It's like homeoviscosity, except instead it's homeostatic control of lipid curvature. So to nutshell this for you, if we take shallow adapted lipids and we squeeze them under pressure, the lipids all become cylinders. And that's a problem because membranes can't be remodeled. You can never get to an inverted phase. And actually proteins don't fit correctly in these membranes either. A lot of proteins, as you may know, are embedded in lipid membranes. So to prevent this from happening, deep sea lipids evolved so that they keep their cone shape under pressure. But no lipids are exempt from the effects of pressure. So if you release the pressure on those deep sea lipids, their hydrocarbon chains unsqueeze, and that should cause the membrane to come apart. So there are some predictions that follow from this hypothesis. For one, we should see that deep sea membranes are unstable at 1 bar at atmospheric pressure. And then two, we'd predict that if we can manipulate curvature in an organism somehow, and it really matters, we should be able to change its pressure tolerance. Prediction 1 reminded us of this experience that might also be painfully familiar to some of you. And that's that when we bring animals up from 1,500, 2,000 meters or deeper, they have this tendency to just melt. And we wondered, could this be because the membranes are pulling apart when they have no pressure? Unfortunately, we don't have a high pressure microscope stage. So we decided to use temperature to cross this phase boundary. And we stained our ctenophore tissue with a dye that only labels lipid membranes. So here, I'm showing you tissue from a species that can be found up at the surface, basically. We collected it in water around 10 degrees Celsius. And as we warm it up to room temp, 20 Celsius, we can see the appearance of a few little blebs. The membranes start to remodel. Let's compare this to a different species, one that is never found at the surface, rarely found shallower than 200 meters. And it never survives at atmospheric pressure for more than a few days, no matter how cold you keep it. We can see that by 10 degrees Celsius, well below room temperature, its membranes are suffused with these little blebs. And then by 20 Celsius, the membranes have disintegrated completely. Because the dye we used only labels membranes, it's reasonable to conclude that melting of these deep sea ctenophores at the surface is a lipid-driven phenomenon. There's also something the color shift of the dye tells us that actually shows that the lipids are going into an inverted phase in this deep sea species. So if you ever wonder why your deep sea animals are melting, hopefully now you have something of an answer. As for prediction 2, well, we didn't have ctenophore CRISPR yet, so we went for it with E. coli. Long story short, we put a gene for PPE production into your sort of ordinary lab rat E. coli bacterium. E. coli doesn't normally make PPE. And then we grew the E. coli in these pressure vessels. So up at the top, we're upper right, we're comparing the growth of a control strain of E. coli, which is shown in gray, with the growth rate of the PPE producing E. coli versus pressure. You've got 1 atmosphere pressure, 250 atmospheres, same as 250 bar. That would be like the pressure at 2,500 meters depth, and then 500 bar, 5,000 meters depth. At 500 bar, the control strain grows about twice as fast, but the PPE E. coli doesn't seem to care. Same goes for survival after it's decompressed. So this is a picture of a plate that we're taking colony counts on. The pressure increases downwards, and you can see that the colony count decreases with pressure for the control, but not for the green PPE producer. We also tried the opposite, replacing all of the PE that's normally in E. coli with a different kind of lipid called PC. PC is actually more fluid than PE, but it's an almost perfect cylinder. And incidentally, a note for later, PC also happens to be the main component of membranes in the shallow Arctic ctenophores we looked at. So things that live around freezing temperatures, but live with 1 atmosphere pressure. And what we see here is that the fluidity improvement that you get from PC is not enough to deal with pressure, because at 500 bar, these bugs are completely dead. And same goes if you look at the survival plate, 0 survival, that's the bottom row. There are no colonies down here. So fluidity alone is apparently not enough to deal with pressure. You need curvature. 3 takeaways from this whole adventure. One is that cold adaptation and pressure adaptation, rather, are definitely different, at least when it comes to membranes. This has some climate change implications. When we look at high latitude shallow organisms, ctenophores at least, like the Arctic ones I was just talking about, we see that their cold water tolerance does not make them pre-adapted to move much deeper as the surface waters warm. In fact, as we just saw in the last experiment, the PC lipids in their membranes would actually be maladapted for that. We also saw that there's some overlap between cold adaptation and pressure adaptation, but for the most part, they're distinct. Temperature drives fluidity-related adaptation, and pressure drives curvature-related adaptation. And now that we know this, we can kind of flip these relationships on their head. In principle, this means that by estimating fluidity or curvature, which we can do based on lipidomics, we can infer organisms' sort of temperature, pressure, happy place and their adaptive potential. Lastly, our successful bioengineering trick with the E. coli opens questions as to how much lipid synthesis genes can tell us about pressure and temperature tolerance through these mechanisms. So let's suppose, generously, that you're hooked by all this and you go submit some of your marine organism samples for lipidomic analysis. Those of you out here who are dealing with marine organism samples. What can you do with it? And I'm basically here to say that if you're interested in the kinds of visualization and biophysical interpretations that you saw here, I'll try to be helpful, and I've posted the code so you can try them at home. So there's a couple of fairly basic software packages I posted on GitHub here. One of them will allow you to visualize lipidomes in what I think is a fairly intuitive way, which is really important because it's such a complex data set. And the other one will allow you to turn these lipidomes into estimates of curvature that you can use to compare between species to try to infer things like temperature and pressure tolerance and differences in temperature and pressure tolerance. If you're interested in these, feel free to ask me about them or just visit the repos and see what you can do with your data. The one on the right, which is for biophysical interpretation, is definitely a work in progress, and you can look forward to some updates soon. For those of you who spotted the marine mammals in the lower right-hand corner and are intrigued, this is part of a project that I have in progress where we're trying to see if the lipids in the brains of diving mammals show homeocurvature adaptation based on how deep they need to dive. So I have some slides on that I'm happy to share, but we're using those, using the tools in that software package to address that hypothesis. I'm also working on new features of the software package that should make it more applicable to bacterial lipids. Things like this really nice data set that's an ISME, and that would make these kinds of biophysical interpretations more applicable to meta lipidomic data, just like metagenomics, where you can measure the omics of microbes in the water column. You can do the same thing for lipids, and you see things like patterns with depth. I'm highlighting one here. There are a lot of people to thank in the various adventures and ongoing projects that you've seen today. I've been privileged to work with experts in all kinds of different disciplines, including deep sea biology at MBARI, lipid biology and lipidomics at UC San Diego, in silico simulations of the way these lipids behave, something I didn't even get into, but again, that I would be happy to address afterwards, which were possible thanks to these experts at UDEL and NIH. A bunch of people who traveled the world to collect some of the animals that you've seen the data from today, the people at the synchrotron, people I'm working with currently on completely different lines of inquiry related to lipids in places like hydrothermal vents. And of course, last but not least, the really hardworking crews of all the ships and vehicles that have given us the ocean access that we need to make this science happen. So many thanks for your time and attention. I'm happy to take questions if we have time for them and there's interest. And very lastly, I will shamelessly plug a 4-minute version of this story that was recently produced by the media whizzes at MBARI, and which I'd be honored if you share with any interested parties. Thank you.
Thanks, Jacob, and great and fascinating presentation today. Very exciting to hear some newer omics methodologies come across our seminar series, and very, very interesting. So we'll go ahead and take some questions now. None yet, but I'll give some others time to think of theirs. I was wondering, Jacob, if you could go into what this phenomena, what you propose it might look like outside of ctenophores. I know you had a quick slide on animals and organisms that traverse temperatures and depths, and I think it's going
to. Did I just lose your audio, Nicole? I can begin by
definitely
answering.
Some pretty fascinating research to follow. That's already, been exciting in its. Can you hear me better now?
Yeah, it was a little bit halting. I got part of the question.
Yeah, my question was, what would you propose these structures how they might change within organisms outside of ctenophores? Like organisms that are traversing depth and pressures, or sorry, depths and temperatures.
Yeah, I think there are two really interesting questions wrapped up in your question. So thanks for asking it. I didn't really go into why we chose ctenophores, but we chose them because they were a good system where we could collect a lot of diversity. That said, the kind of membrane lipids that ctenophores make, like the basic shape of the molecule, it's the same as in all other animal. So we have no reason to think that the same thing wouldn't be happening in different kinds of animals. And that is exactly where basically all of my ongoing research is pointed. We're trying to figure out now exactly how widespread is this kind of adaptation, because that would really help us inform. Well, it tells us more fundamentally about how life works in the open ocean, and it could also tell us about how different kinds of organisms might respond to things like surface water warming, and how it might affect things like their vertical migration. The second part of your question that I thought was really interesting is you're asking about animals that migrate up and down in the water column, which is a lot of people here probably know many open ocean animals do every day. Ctenophores don't really. They tend to hang in the same depth band, but a lot of animals go up and down every few hours. And frankly, we don't know yet how those organisms deal with this effect. One hypothesis is that they remodel the shape of their lipids, basically to change the curvature of their lipids as they migrate, which would mean that they're doing the biochemistry very quickly. But we don't have the data to tell yet. So I look forward to sharing that once we figure it out.
Thank you. Thanks. And yeah, step by step. Very, very exciting research. Thanks for that. Okay, we have one question in from Allison, who asks, how did the collections at depth take place to ensure the change in pressure didn't confound results?
Really good question. Really good question, totally tied to your question, Nicole. I will show you really quickly how the collection took place, just for those who are curious. We collected most of the really deep water animals in these jars called detritus samplers. Can you all see this video?
Yes.
Yep, there it is. And so we have these really talented pilots who can catch these things in jars. To answer Allison's question though, these jars do not retain pressure when you bring them up. The pressure changes. And frankly, sometimes when we would get these animals to the surface, they would have melted. That would happen. The thing is, as far as we know, they are not capable of changing their lipid composition that fast. And I actually have some data that I can use to speak to that. That I think is kind of cool. But they can't change their membrane composition that fast. So the idea is that even if the animal is melted because of the stress of its being depressurized, its biochemistry is not going to change that fast because its enzymes don't have time to remodel the lipids. And we still have to allow for the possibility that the animal could be actively trying to respond to that decompression stress and remodel its lipids as the ROV, as the sub moves towards the surface. But even if that was happening, we still saw these differences that were correlated with depth by the time we recovered the animal back on board ship. So we, in response to your question, Allison, we can't really deal with the stress of decompression and its effects quantitatively, but I think the differences that we saw between the species are still real. Because they represent whatever's left of the difference in their lipids by the time you get the animals back. We also have some data showing that at least in one species of deep sea ctenophore, their lipid makeup is hardwired. And this might come from the fact that they basically live at the same depth or within the same depth range their entire lives. I think this has some really interesting implications for like the adaptability of membranes in a changing ocean. Basically, we had some of these animals that were collected fresh. We had 3 more of these animals that were kept in the Monterey Bay Aquarium for 3 months. They're the first aquarium to be able to keep these alive. And the crazy thing is that their lipidomes after 3 months are identical, no significant differences. So you can see that on average, the size of these color blocks are all the same. If you look at the distributions of their hydrocarbon chains, who's this here? These are the columns are different head groups here. And they show the top plot is the number of carbons in the chain. The bottom plot is the number of double bonds, kinks in the chain. The vertical dashed lines show the averages. The averages for fresh out of the deep sea versus sitting in a tank at 1 atmosphere for 3 months are exactly the same. So we think some of the lipids are hardwired. They don't change when we recover these particular animals. If we were dealing with vertical migrators, it might be different. Thanks for humoring me, Nicole.
Great explanation for Allison and a little bit more detail for the audience. Our next question is from Zach, who says, amazing talk, Jacob. NOAA is particularly interested in understanding how organisms are responding to ocean acidification and temperature changes associated with climate change. Do you have any thoughts on whether changing pH would impact lipid physiology? And what are the large-scale implications here based on lipid physiology with a warming ocean?
That's a really good question, and to my knowledge, a very open question. Lipids is like we saw a while ago, the heads of lipids, they're in direct contact with water. And some of them have structures in their head group here that is responsive to pH. It can get protonated or deprotonated. So we know that there are certain lipids whose shape and whose behavior is responsive to pH. Do we know that much about the effects? Do we know as much as we do about the effects of temperature or pressure? No, not really. That's one of the foci of my current research that I'm doing at hydrothermal vents. And my hunch is we may have to look at organisms that are adapted to really extreme pHs, like not ocean acidification pHs, like hot spring acid pHs, things like that, to understand what kinds of adaptations take place. But once we study those extremophile organisms and understand the adaptations, we might be able to understand the sort of effects that that ocean acidification is going to have on the membranes of animals, more broadly in the ocean. It's all a matter of figuring out structure function. So basically, the story I told you today, was about how the structure of these molecules affects what the organism can survive. And we just need to establish more of those links. The more links we can establish between the structure of the molecule and the function of the organism, the more we can use omics to tell what the organisms can take, what they can adapt to.
Awesome. Thanks, Jacob. Alrighty. Our next question comes from Stephanie, who asks, are there any other small molecule or unique enzyme adaptations that you found in your work, or any other hypothesis? What are future directions for this project? I know we covered them a little bit prior, but if you could elaborate a little bit more.
No, I appreciate that. That's a great question. And I'm actually really glad you asked it, Stephanie, because the last takeaway I would want anybody to have from this is that lipids are the only thing that matter for temperature and pressure adaptation. I would contend that they matter a lot, but when you think about a cell, it has a bunch of parts. It's got the membrane, a variety of proteins and enzymes, a bunch of other small molecules, and they all have to work right for an organism to have fitness to survive. So long story short, yes, there are other things that matter. Other people have found some of them. I have found some in my past research. It seems like the next most pressure sensitive thing, at least, behind lipids and membranes, is proteins. If you think of the reason lipids are pressure sensitive, it's basically because different parts of the molecule are more squeezable. So the comparison I like to use for deep sea people here is that the hydrocarbon chains of the lipids are super squeezable. They're like a styrofoam cup, and if you take a styrofoam cup to the bottom of the ocean, it gets squeezed. The top is something incompressible, more like an aluminum can, and so when you apply pressure, the shape of the molecule changes. Proteins are the same way. Some parts are more squeezable than others. So you can actually pick up signatures of deep sea adaptation in the genomes and transcriptomes that code for proteins. But I would argue that it's a little bit harder to do. And then there's another molecule that's really famous for helping organisms with high pressure adaptation, and it's called TMAO. And that's a type of small molecule that they call an osmolyte. Basically what it does is it arranges itself around the proteins and helps them fold correctly, even when they're under high pressure, prevents them from getting squeezed out of shape. So long story short, no lipids are not the only thing that matters for temperature and pressure tolerance, but they matter a lot. As far as future directions go, right now I'm really focused on figuring out how universal these principles are. If we can demonstrate that this happens not just in ctenophores, but in other kinds of animals, if you want to think of the most different animal you can, think perhaps of diving marine mammals. If it happens in other animals, if it happens in bacteria, if it happens even in archaea, a different domain of microbe, then we have some really powerful universal principles that we can use to predict functional behaviors from omics data. So right now, I'm just doing a lot more of these comparative studies in very different types of organisms, just to see how far we can push this homeoviscosity principle and the homeocurvature principle.
Well, thank you very much, Jacob. I think we'll end on that. Very exciting directions for your research, and as well, exciting publications as well of this most recent project. With that, thank you, Jacob, very much. And thank you, everybody, for joining today. Our next seminar series will be Wednesday, February 22nd by Anita Ray, who will speak on cryptic species identification. With that, we'll see you guys next time. And thank you, Jacob, so much for the presentation today.
Thank you all, and thanks for the great questions.